Synthetic biology is a new and emerging area in science that asks the question, can we engineer biology reliably? So think of an engineer building bridges out of nuts, bolts and girders to make these large structures that we can see, and translating that right down to the submicroscopic scale of molecules and trying to build things with molecules.
We are engaged in a big experiment here. We're starting to do new things with science. New questions are being asked and different collaborations are being engaged in new ways. That's an experiment. It's an exciting experiment.
We're bringing together physicists, biologists, chemists, mathematicians, engineers, and that's quite brave. It's quite difficult because we all speak different languages. So the first hurdle we have to get over is learning to speak with each other.
I'm here to learn about synthetic biology, this new subject, and the discussions we have are quite astounding. They range from completely imaginative ideas, which may never work, to things which are very, very grounded, and we could probably start developing them tomorrow.
The sharing is so extensive, actually, that there are real discussions going on. People feel really free to discuss their ideas here.
It's important to engage with new people in the field, young people, because they have they're not so restricted by the old ideas. You know, they have no idea of what's impossible.
Synthetic biology may be taking us into a new and exciting future in which we can engineer biological systems to do new and useful things. Produce drugs, produce biofuels, produce biomaterials for medicine. That's the potential. The question at the moment is, do we understand enough about biology to engineer it?
In synthetic biology, we've got loads of scientists working beyond the boundaries that they've previously worked. And by their own admission, they're going to have lack of knowledge about certain components. They are the first to freely admit that they're dealing with disciplinary boundaries that they haven't crossed before. They're the first to admit that they have gaps in their knowledge. And the whole point of a network like the Synthetic Components Network is to bring people from different disciplines together, to plug those gaps in knowledge and to get those new collaborations going.
People in synthetic biology are engineering DNA, other people in synthetic biology are engineering whole organisms. We're somewhere in the middle, we're trying to engineer the workhorses of biology. They're called the proteins.
One of the proteins I always find quite magical is a protein called green fluorescent protein. A linear sequence of amino acids folds into a protein. One would argue it's a bit like a car building itself. That's how proteins fold. It uses those individual components to generate that three-dimensional structure. Thinking about that always marvels me. While we use the term synthetic components, they are essentially molecules that nature already uses to carry out most of its processes. It's just the synthetic parts are influencing them by changing and modulating them directly.
So one of the things that we're hoping to do in the Synthetic Components Network is to create a set of biological molecules that we really understand. We understand how these are built and how they work together, how they assemble into larger, more useful biological structures, from the bottom up to try and engineer things reliably.
From the engineering side, I think it's very exciting. These components, these new parts, give us the ability to make things we've never made before. You take these components, which are components at a tiny, almost invisible scale. They're biologically derived or biologically inspired, and we have this box of Lego that the best engineers can come along, can pick the bits that we're interested in, put them together with a goal in mind, and create something which is much greater than the sum of the parts.
When I was a kid and you had Lego, it was just simple Lego bricks and you built something. When my kids were growing up playing with Lego, you bought a Lego helicopter, which you built as a Lego helicopter, but the little design was there to show you could make a Lego racing car out of the same bricks, because you knew how to put the bits together. So if we can take bits of a cell apart and work out, yes, they fit back together like this to work, but actually if we take those bits and that bit from over there, we could make something else, and that could be really useful. Who knows for what? So my name's George Banting. I'm from the Biochemistry Department at the University of Bristol.
I'm Steve Evans from the Department of Physics and Astronomy in Leeds. And what got me interested in membranes as a physicist was that it acts as this interface between the interior and the exterior of the cell, and almost all of the interesting processes have to somehow take place across that membrane. I suppose if I was trying to give an analogy to somebody, then it would be very much like the molecules that form a soap bubble. So everybody knows what a soap bubble is, and if you blow a soap bubble, you get this beautiful shape. And this is formed of molecules that are not very different from lipid molecules that form the membrane.
You can think of the membranes as being devices which separate one part of the cell from another part of the cell, and in the context of the cell, they're the device which defines the cell as being an entity and separates it from other cells. And I think the importance of it is borne out by the fact that from the prediction analysis of the human genome sequence, the suggestion is that a third of all proteins encoded by the human genome are membrane proteins. So that itself says that this is an extremely important area, so the more we can understand about that interface, the better.
For me, starting with a membrane is the simplest thing I can think of. Make a liposome, it's empty, there's nothing there, just a shell. But then can you start to go on and increase the complexity of that? And I think that's what we hope to do in our collaboration. So having the membrane proteins in these artificial constructs gives us a platform for being able to study these membrane proteins in a way that people can't easily do at the moment. It's postulated that 50% of all drugs that will be discovered in the next 10 years will act on membrane proteins.
Synthetic biology touches on real issues of substance. Synthetic biology promises the capacity to engineer life from the ground up. Now, as a social scientist, I want to explore that question more carefully. What does that mean?
We're talking here about a group of scientists who have been working incredibly hard for quite a long time, actually. And then suddenly the whole world is interested in what they're doing and thinks that what they're doing has got quite profound ethical implications.
We actually need a new way of thinking about this that enables us as a society to think in a more intelligent way about the way in which we innovate as a society, and to kind of, in a way, learn to deal with uncertainty in a new way.
It's not necessarily a problem as long as A, we all understand generally where the field is going and what implications it may give rise to, and B, we also understand some of the core concepts and we can find a common language to talk about it in that way.
And if these initiatives that bring together scientists and social scientists together with the public are a way of, in a sense, creating a new way of thinking about this, a new way of developing a conversation, I think that's great, and I think that could be really worthwhile.
The reason synthetic biology is an experiment is because we don't understand enough yet about biological molecules and how they're pieced together, how they work together in order to engineer them reliably. So ideally, synthetic biology will be an engineering discipline, but that must be underpinned by understanding of biological molecules in order to be able to engineer them.
I just think it's amazing that we can look at nature and be inspired by the things that they can make. And these incredibly simple organisms control and produce materials that we can't produce ourselves with all of our technology.
We want to make sure that this technology is developed in as safe a way as possible, that the technology isn't grabbed by commercial companies such that it can never be used in developing countries to help people who really need it. And also that in the sorts of things that we're choosing to develop, we're doing so in a way that is perhaps targeting the most pressing problems in the world.
If we want to build amazing future technologies for the human race, we are going to do it in the end by having understood how biology works, how nanotechnology works at the fundamental level.